Hello, we're Links China, a high school iGEM team from Shenzhen, China. Our 15 team members are located from high schools around the world through Links Academy located in Shenzhen. And today we are here to present you our project Pikachu, Peeling Constructed Nanowire Rechargeable Humidity Battery. So in March, our team was ecstatic to find a Nature article published that describes a way to use G-sulfur reducens pili to extract nanowire film and produce a stable current voltage. It suddenly occurred to us that it would be extremely exciting to make a bio-battery that can recharge itself by simply using humidity from air. So how does it actually generate electricity? It seems quite bizarre that it can simply extract energy from water in the air. In the explanation given by the article, it says there are functional groups, carboxyl groups, on the surface of the nanowires. And these functional groups will interact with water molecules, transmitting the H protons down the gradient during the diffusion process, forming a water gradient. And this will produce the continuous voltage output, and the functions are over an extended period of time. There are two electrodes, one on the top and bottom, both are gold, and they would connect with each other, making sure that the electrons are transmitted and the process circulates. However, instead of using G-sulfur reducens, which are hard to maintain and to transform in a limited lab setting, we decided to use the lab-friendly E. coli instead to produce the protein nanowires we need for bio-battery production. Especially for the K12 strain, it's possible to make much larger production, and also it's easier to make modifications within. So what exactly are these nanowires? These are actually type 4 peeling, and are fibers anchored on the surface of bacteria that help adherence, mobility, and transport functions. Bacteria assemble peeling with its internal protein assembly system is shown on the right of the slide. And it's pretty much contained in a lot of strains, including E. coli and other bacteria. However, G-sulfur reducens are one of the few that actually transmit electricity, or called E-peeling. But the rest has some of the functions but do not actually show characteristics of transporting electrons. Now let's take a closer look at the peeling assembly structure. So it's mainly consisted of 4 parts: the minor peeling, major peeling, assembly platform complex, and also the ATPase. So imagine this: the major peeling would gather from around, and slowly, guided by the minor peeling, it will extend itself. And like knitting, it will just go longer and longer, then gradually secreted by this structure. Now that we've learned about the theoretical part, it's time to learn how to produce the peeling. Because the genetic circuit for making the nanowire is quite complicated, we decided to make it into 4 smaller parts. The first one with the vector PET28A, the second one pill A and half B and C, the third one half MNOPQ, and the fourth one PBDA, B, YGDB, and PBDC. We successfully assembled the DNA and confirmed it with the electrophotogram results. The first one for pill A, the second one for PBDA to C construction, and the third one as shown is half MNOPQ. There are sequence differences on the generator between strain DH5A and DHL933. So to make the generator functional with the point mutation shown on the right, at these sites to derive the sequence in EDL933. So we successfully assembled the nanowire plasmid, but is it working? So after constructing the plasmid, we directly came into pillar production and purification at once to confirm that the plasmid is functional. We first cultured E. coli with the nanowire plasmid in some M9 medium for harvest of a large amount of source of pili. Then we collected the bacteria from the plate and then used the blender for extraction of the pili. Further, we infused the liquid with pili into the spinning filter for pili purification. The residue on the membrane would be the pili. Then we use SEM photography. It is basically like using a microscope zoomed in to see the nanowires. So these are the ones that we've purified. On the graph A, it is with a lower magnification. It shows that the pili are evenly distributed. On the right side, in graph B, it shows that there are porous structures between each pili, meaning it is beneficial for creating a humidity gradient along different depths of the pili located. To confirm that the things we produced are pili and not actually anything else, we used Western blot for further confirmation. And the slides shown here are the typical steps of a Western blot experiment. For Western blot, we added his tags on the pill A sequence, or if there exists to be pill A protein, then it will successfully combine with the antibodies and the secondary antibodies. So for our result, it shows that we have succeeded. It is positive, showing stripes between 10 and 20. This means that the production of the pili is successful. So after confirming that we actually made the pili successfully, how do we actually make it into a battery? From the example given by the Nature article, they used electrodes, gold stripes, on both ends of the nanowires. The top one, a smaller one, and the bottom one, a larger one. And for us, we decided to use carbon nanotubes or graphene to use as the electrodes. So the process is mainly like this. We will first drop the nanowire directly onto the electrodes, and then when the solution is evaporated, it will be left with a single film of nanowires. And the picture shown on the right are the samples of our own nanowire batteries. So for our own batteries, we made our first nanowire film sample several weeks ago, which has a substantiation of producing a fluctuating voltage. We also measured the resistance and calculated related parameters.
哦，不对，那是
610
610毫伏。
produced several other prototypes.
0.06
We produced the film into PCR tubelets and connected them with wires in order to connect the film in series to produce a higher voltage. However, we have yet succeeded because of the short circuit and some other issues. This sample is not very precisely produced, since the nanowire solution was just simply dropped and dried on the glass side. We have several goals. First, we need to connect several films in series to produce enough voltage to power specific electronic appliances. Secondly, we need to do more testing to determine the relationship between ambient humidity and voltage produced.
它是这样的，因为这个很容易戳破。
对，所以说
这东西很容易戳破。
对。
如果它不戳破的话，
但是明显碰到的话就会数就变大，很明显。
很明显，你碰到它的表面，它
你继续夹那个，我把那个继续夹那个，按照刚刚的方法，我把这个滴点水试试。
滴水没有用吧？
为什么不是没有用？它这个有个浓度差了，主要就是靠滴很少一点水，然后继续录啊。你录了吧？啊，好。我还以为没开始。
可是没开始。
上次就有3开始。
来，
滴一下水。
滴水。我觉得跟水没有关系。
滴水的话，按照理它会高一些。
为什么现在它不是很稳呢？
这一般应该不会很稳。
好，来。滴水有电压。
有。这是负的吗？但是。
对呀。
负的电压吗？哦，对。
70。负的70。
哦，是因为反着拿是吧？
滴水也有电压，为什么？
正常不会。
负暗示，它电压会越来越小。
会吗？
你看。OK，
听。
After making our battery, how do we actually apply it? And since our battery is very small and convenient to be carried around, we thought of providing electricity for small devices, and especially the tracking devices for migrating birds. This occurred to us, for Shenzhen itself is a natural habitat for thousands of birds, and they are an essential part of the ecosystem. After we provide electricity for the GPS system on the devices, it can be carried around to anywhere around the globe, through rainforests to through the desert. Because it only uses humidity from the air, it can always recharge and repower itself. And in August, we had a meeting with the natural reserve in Dapeng, Shenzhen, and they gave us a lot of useful suggestions for how we can actually use our devices. Along with that, for public education, we started making a game called iGEM, which is basically an escape room game in a laboratory, and the protagonist needs to explore the equipment around and try to escape. Also, we are trying to upload a lot of vlogs onto Bilibili and YouTube, and we are also trying to publish articles on social platforms as well as lectures. This is our project overall, and thank you for listening.